Good morning, students. This is your teacher Neela Mehlawat, and today we'll start a very interesting, very pleasant, and very, you can say, enthusiastic and highly a topic that will create a lot of curiosity in us. See, genetic itself is a very, inquisitive subject. And humans also, they are curious to know each and everything around us. So we'll start with this topic today. So you be ready. You take your books and notebooks so that we can start. Right, everyone? And I hope you're keeping safe. You are maintaining the social distancing and keeping your family and everybody safe in your surroundings. Okay, everyone? So shall we start now? Let's start, everyone. Now, before we actually come to the topic, we need to understand. See, genetic code we have to talk about. Genetic code. How information is being coded, isn't it? That's a very important one. So we must understand, see, three processes we have to talk about. One is DNA can produce DNA. And this process we call it as replication. And we have learned this also. Isn't it? DNA can produce DNA. This process we call it as replication. It's a very important one, right? Then from DNA, mRNA will be made. And this process of making mRNA from DNA is called transcription. Right, everyone? This process is called transcription. And the third process where mRNA will help in the production of proteins. This process is called translation. Okay, this process is called translation. So we have three processes in series. One is replication, another is transcription, third is translation. And all the three, they depend on each other. So replication we have spoken about, we have read about it so far. Transcription we have just finished. Now we were left with translation. Clear? Now before we come to the translation, we must understand that mRNA has some information. Right? mRNA has got some information from DNA. And this information will help in generating the proteins. So we must understand how this information is present in RNA. What is the format of this information? Like suppose if we have started with the topic, you will have your books. Isn't it? From book you will be reading or you will be seeing or checking, and in the notebook you will be writing. So we need to understand that how the information is present in mRNA. Where is this information written and how this information is written and how this information will be used to form the proteins. So that is the question we'll be dealing today. And that is the question where the genetic concept of genetic code is there. So before we start with translation, we need to understand the genetic code. Now see, there are 4 bases. Okay. Adenine, guanine, cytosine and uracil or thymine. In case of DNA, we have adenine, guanine, cytosine and thymine. While in case of RNA, we have adenine, guanine, cytosine and uracil. Right? So we have 4 bases. So your DNA and RNA both are constituted by the 4 nitrogenous bases. Coming to the proteins, there are 20 proteins, or you can say 20 amino acids in general. AA means amino acids. And I've listed here those 20 amino acids: arginine, proline, lysine, histidine, glutamic acid, aspartic acid, tyrosine, threonine, valine, cytosine, glycine, methionine and leucine. So we have 20 amino acids. Just imagine. And we have only 4 bases. So how come the 4 bases can produce 20 amino acids? This is a question. And we must understand देखिए हमारे पास 4 बेसिस हैं और 4 बेसिस के पास कैसे इंफॉर्मेशन है कि हमारे 20 अमाइनो एसिड्स प्रोड्यूस हो रहे हैं. वी गॉट टू सी दिस कॉन्सेप्ट. नाउ लेट्स सी how amino acids will be formed. Suppose we have 4 bases A, G, T and C. Now suppose we are assuming that one base will form one amino acid. Okay, one amino acid will be formed by one base. So we have how many bases? 4 bases. So 4 bases will form how many amino acids? 4 amino acids. And in totality, how many amino acids we have? We have 20 amino acids. So this concept is failed. So it will not work because one nitrogenous base cannot form all the 20 amino acids that we have today. So let's see. Let's assume that 2 bases will form 1 amino acids. Let's have this concept. So 2 bases will form 1 amino acid. Now if we go by this concept, okay, so any 2 can be taken at one point of the time. We can take Ag, we can take GT, we can take TC, isn't it? Any 2 bases at a time can form amino acid. So if we go by this concept, then in that case we have 16 amino acids possible. Okay, अगर हम 2 bases का concept लेते हैं तो 16 amino acids की formation possible है. And we have total 20 amino acids. So even this concept will also not work. Now there was a scientist named as George Gamow. He argued, George Gamow, understood? He argued that there will be 3 bases that will code for one protein. Okay, 3 bases will code for one protein. I hope you've understood. Let's see this. How 3 protein can code for a base. He said, let's go by his concept. We have 4 bases A, G, T, C, isn't it? We have 4 bases A, G, T, C. Now he said that 3 bases will code one amino acid. So we can have any, isn't it? He said 3 bases will code for one amino acid. Now any 3 base can be used. Now here we have 4 bases. Now let's suppose I have 3 positions possible, right? Because 3 bases have to be taken at one point of the time. So I can take any base on the first position. Suppose I take A. So I have 4 possibility on the first. I can have A, G, T, C. Second again I can take any C, suppose I take. So I can have out of 4 any. So again I have 4 possibilities. Suppose here also I can take C again. So again at this position, first position we have 4 possibilities. Second position we also have 4 possibilities. Third position also we have 4 possibilities. So in total, just multiply this. 4 into 4 into 4, how many? 64 combinations. So how many combinations? 64 combinations, right? So as per the George Gamow, he said 3 bases need to be present to code for one amino acid. And if we follow this, there are 64 combinations which are possible. So he said that 20 amino acids are produced from 4 bases, means at one point of the time 3 bases will be together, and these will generate 3 combinations. And since there are 3 possibilities, 3 bases, this will be called a codon. ठीक है, क्योंकि हमारे पास 3 बेसिस हैं तो इन 3 बेसिस के कलेक्शन को हम कहेंगे कोडोन. तो हमारे पास कितने कोडोन हैं? टोटल 64 कोडोन्स पॉसिबल हैं. ठीक है, क्लियर? लेट्स कम टू द नेक्स्ट कॉन्सेप्ट. सी, डीएनए से हमने एमआरएनए बनाया. एमआरएनए से हमारे प्रोटीन्स बनेंगे, यानी अमाइनो एसिड्स बनेंगे. एंड जॉर्ज सेड दैट 3 bases will make 1 amino acids. So let's see, this is a sequence which is there. Okay, mRNA has a sequence A, T, G, C, T, G, A, G, G, C. So 3 will make 1 amino acid. These 3 will make 1 amino acid. These 3 will make 1 amino acid. Okay, So as per George, he said 3 bases will code for 1 amino acids. So we have in total 64 combinations, 64 codons. This single is this 3 bases will form a triplet. So we call it as triplet codon. Okay, this is called as triplet codon. Okay, clear? Now let's define what is a codon. So codon, genetic code, is a dictionary. Like we have an English dictionary where we can consult the meaning of English words. Same way, genetic code is a dictionary, isn't it? And it is made up of genetic words, and these genetic words are called codons. And these codons have the information to make a amino acid. Clear? And each codon has 3 bases. That is why we call it as a triplet codon. Okay, so the codons, they are triplet. And in total we have 64 codons. We have 64 combinations. 61 codons will code for the amino acids, while 3 codons do not form any amino acids. So total we have 64 combinations. 61 will code for amino acids and 3 will not code for any amino acid. Clear? Now there are 2 types of codons which are possible. One is a sense codon, another is a signal codon. So 2 main types of codons are there. Sense codon, signal codon. Sense means each codon will have a meaning. Each codon will have a meaning. Each codon will have a specific meaning. Signal codon means they will have the message, and that message will be conveyed or processed. Okay, sense codons means they will have a definite meaning. Signal codons means they will have some message, and that message is very, very important. Clear? And these signal codons, they are of 2 types. One is start codon, another is called stop codons. Start codons, they are also called as initiating codons, right? S-T-A-R-T, start codon. Clear? Let's write it again. Start codon. So your initiating codons, again I have written the wrong, sorry, start. Okay. So initiating codons are also called as start codons. And as the name suggests, they will start the process. Okay. Initiating codons, they will start the process. So AUG is your initiating codon. Always the process will be started by AUG. And this AUG produces an amino acid named methionine. ठीक है? So initiating codon को हम start codon भी कहते हैं, और हमारा start codon होता है AUG. और AUG हमेशा methionine amino acid बनाता है in all organisms. So सबसे पहले कौन सा प्रोटीन, कौन सा अमाइनो एसिड बनता है? methionine. This is the first amino acid which is made. Then second is terminating codons. Terminating codons means stop codons. Okay, terminating codons means stop codons, means they will stop the process. So there are 3 codons. Okay, UAA, UAG and UGA. These 3 codons, they are called termination codons. They are called as stop codons. They will stop the process. And they're also known by another word that is called nonsense codons. They're also called as nonsense codons. Okay, dear students? There is one more term that you will find generally in genetics, which is called anticodon. Anti means opposite. Okay, anti means opposite. So codon के opposite. So हमारा codon जो होता है वो mRNA पर होता है. mRNA has a codon and tRNA has an anticodon. ठीक है? So DNA has made mRNA, and this mRNA has some information, and this information will be read by tRNA. ठीक है? So DNA से mRNA बनाया हमने. mRNA में इंफॉर्मेशन है, कुछ मैसेज है, उस मैसेज को रीड tRNA करेगा, राइट? सो tRNA कैसे रीड करेगा? tRNA will show the anticodon. सो टीआरएनए पर एंटी कोडोन होता है, एमआरएनए पर कोडोन होता है. कोडोन के पास इंफॉर्मेशन है एंड दिस कोडोन विल बी रेड बाय द टीआरएनए. और टीआरएनए पे जो इसके कॉम्प्लीमेंट्री बेसिस बनेंगे उसे हम कहेंगे एंटी कोडोन. वी नो डियर स्टूडेंट्स, or A will always connect with T. That means adenine will always show connection with thymine. G will always connect with C or U. Okay. So U is here, you will have A here, C is here, G will be here, U is here, or G is here, C is here, G will be here. So anticodon is always present on tRNA. So tRNA अपना एंटीकोडोन बनाता है जो कि कोडोन के कॉम्प्लीमेंट्री होता है, जो उस इंफॉर्मेशन को रीड करता है जो एमआरएनए के पास है. क्लियर है? आई होप यू अंडरस्टूड. यस, लेट्स गो फर्दर. लेट्स हैव डिफरेंशिएट अगेन कोडोन एंड एंटीकोडोन. फ्यू मोर डिफरेंसेस. Codons are always present on DNA and RNA, and anticodon is present in tRNA, isn't it? Just now we've talked about tRNA. It can never be present on DNA. Codons, they are always read from 5' direction to 3' direction, while anticodons, they are read from 3' to 5'. So this is a basic difference. Clear? Understood everyone? Third is, codons have a specific sequence in nucleic acid. Anticodons, they are always present in cells. Okay, codons have a sequence present in nucleic acids, while anticodon, they are present in cells. Codon will define the anticodon. Codon के opposite anticodon बनाया जाता है. Clear है? Anticodon will bring about the synthesis of amino acid. So anticodon जो है वो information को read करता है जो mRNA के पास होती है, and then only a particular amino acid will be made. Okay dear students? I hope the concept is clear. The introduction of genetic code is clear. तो जो भी information mRNA के पास मौजूद होती है उसे tRNA रीड करता है. और कैसे रीड करता है? एंटीकोडोन की हेल्प से. ओके, सो एमआरएनए हैज इंफॉर्मेशन, दैट इंफॉर्मेशन इज decoded or read by tRNA. And how this information is there in mRNA? It is always in the form of codons, in the form of triplet codons, right? I hope you understood. If there is any query, any doubt, you can ask. I'll be eagerly waiting for your questions. Right, everyone? Okay, so this will be your today's homework. You will make notes of this topic, and then you will read the page numbers 111 and 112 and underline the important terms. Make 1-mark questions from this particular topic. It's a very encouraging one, very interesting one, right? And it is 100% asked in the exams. Very important also from the board point of view. And even from the competition point of view, because you'll be appearing in number of competition exams. So from competition of viewpoint also, this topic is very, very important. Okay, everyone? Okay. So bye, everyone.